We are at the Institute of Organic Chemistry and Biochemistry of the Czech Academy of Sciences, and my group is called Drug Discovery. I am Pavel Mayer, the head of Drug Discovery Group. Nature of our science is very diverse. We have many projects because we collaborate with many groups within the institute, and also we have many outside collaboration. Our focus is on medicinal chemistry with the final goal and aim to discover drugs, to introduce something to the market. What I think is what we bring to the ATRIS project is experience in both organic chemistry and medicinal chemistry. The difference is that even a great organic chemist makes a very interesting molecule, but the molecule may not be suitable for biological activity. So one of the projects I would like to mention that was supported by ATRIS is glutamate carboxypeptidase. This is a funny enzyme because it has two very different, or actually several very different roles within our body. We try to inhibit it for several different reasons. One of them is to get some neuroprotective compound, which is very challenging because the inhibitors are very polar compounds, and they very difficult is to cross this barrier. And another use of inhibitors of this enzyme are in therapy of prostate cancer, because it is actually originally it had two names, and one of them is prostate-specific membrane antigen. Inhibiting this activity can be, or targeting this activity on the surface of prostate cells or prostate cancer cells that originated from prostate, can actually treat the tumors. Now the problem is that this enzyme is not only in the prostate, not only in the nerves, but also in our salivary gland and in our kidneys. So this so-called radiotherapy of prostate tumors is actually also damaging salivary gland and kidney. And so we have a compound that specifically protects salivary gland and kidney partially from binding of the radioligand. So the radioligand then destroys the tumor, but not the salivary gland, not our kidney. Another project I would like to mention that we did within the ATRIS funding is itaconic acid. Itaconic acid is an immunomodulator, and the disbalance of this immunomodulation leads to so-called alopecia areata. So we have a prodrug, which is a modified itaconic acid that releases the itaconic acid on the site of action, and this compound should help in regrowth of hair, and is currently in a clinical trial in a company Spark that, crossing fingers, hopefully will hit the market and help people with this problem. So yet another project is decitabine prodrugs. Decitabine is a compound that is being used to treat myeloid leukemia. It is quite unstable. It must be handled in a very special way. And so what we did, we modified the molecule of decitabine to be more stable and to extend use to other indication. But now we started a collaboration with folks at Biochemistry. Hopefully, if everything goes well, to start a startup.
Our typical collaborators are scientists from many institutes in our country, from universities in our country, and also many scientific institutes from various countries in Europe and from USA. For example, our, I would say, best collaborator is Johns Hopkins University in Baltimore in USA, the team of Professor Barbara Slasher. And we have also a very interesting collaboration with Israel, with Weizmann Institute, and I would say also very nice and very important collaboration is with companies. For example, Gilead Sciences in California in USA, with Rega Institute of Medical Research, and Several years ago, we had a very nice collaboration with the team of Professor Jan Balazzarini and Graziella Andrei on compounds targeted to DNA viruses. These compounds were produced in our laboratories. Also, this research was supported by ADRIS grants. We have several very nice publications with them.
So one of the major benefits being part of ADRIS network, of course, is a network, are the people we collaborate with, because we are medicinal chemists, but medicinal chemistry without testing that continues on these institutions is absolutely essential. So that is definitely very important for our work to make sense. And of course, we are grateful for the funding that ADRIS provides for us, because without funding, of course, there is no science.